Great. This project was on the effect of different fertilizers on tomato plant growth, and my name is Hailey Haas. The purpose of this project was to compare the effects of fertilizer high in nitrogen and fertilizer high in phosphorus on a tomato plant's growth. The fertilizer high in nitrogen was a blood meal, and the fertilizer high in phosphorus was a bone meal. The control group was a tomato plant grown without the use of any fertilizer, so that the other two plants could be compared to it. The plants were planted in small pots and grown indoors. They all received the same amount of soil, water, and sunlight. The hypothesis I came up with was that if fertilizer was higher in nitrogen than phosphorus, then the tomato plant will grow faster in height. The hypothesis was not supported by the results, and the plant with the higher phosphorus fertilizer grew faster. Based on these results, I can conclude that phosphorus is better for tomato plant growth. This means this fertilizer is most likely better for gardening and vegetables. The three plants were measured over a course of 24 days. The data shows that the tomato plant with more phosphorus ended up with a faster growth rate than the control group and nitrogen group. The plant without fertilizer germinated faster and was taller than the other two groups for the first 21 days. A scientific journal written in 2010 stated a biofertilizer such as plant growth promoting rhizobacteria, also known as PGPR, prompted more microbial activity, and the cycle of events made more nitrogen available for plants' uptake. It was a study conducted to find better options to promote the nitrogen levels in plants than nitrogen fertilizer, because it is bad for the environment. This study helps support my hypothesis that there are better fertilizers to promote tomato plant growth. The three main research questions for this experiment were: how does fertilizer affect tomato plant growth? What kinds of fertilizer produce the fastest growth and height? And is nitrogen-rich fertilizer better than phosphorus-based fertilizer? Methods. So the materials included bone meal, blood meal, potting soil, water, and tomato seeds. I filled 3 different pots with 1.5 cups of potting soil. I planted a total of 9 tomato seeds, 3 in each pot. The seeds were covered with about 1/4 of an inch of potting soil. One of the pots, I added a tablespoon of the bone meal, and this was the fertilizer high in phosphorus. And then to another one of the pots, I added a tablespoon of blood meal, and this was a fertilizer high in nitrogen. The third pot was not given any fertilizer. Every 3 days, measurement of height was taken, and the plants were given a tablespoon of water. They were grown inside and received the same amount of sunlight. The results show that the tomato plant that received the phosphorus fertilizer had more growth than the plant without fertilizer and the plant with nitrogen fertilizer. The plant without fertilizer was taller in height for the first 18 days. The plant with the phosphorus fertilizer was then measured taller than the no fertilizer plant on day 21. The plant with nitrogen fertilizer was the shortest out of the 3 plants. Table 1 shows a chart of the measurements of height of the plants over a course of 24 days. As shown, the plant with the phosphorus fertilizer presented the most amount of growth at the end. The plant without fertilizer showed more growth than the plant with the nitrogen fertilizer. Table 2 compares the nutrient element ratios of the fertilizers. This includes the nitrogen, phosphorus, and potassium percentages. The blood meal has 12% nitrogen, 0% phosphate, and 0% potassium. The bone meal had 4% nitrogen, 12% phosphate, and 0% potassium. I would also like to note that it has a 12% of calcium. Figure 1 shows the growth of the tomato plants in height measured in centimeters. The orange line indicates the control group, or the plant that was not given fertilizer. The dark blue line is the growth of the plant given bone meal, and the light blue is the plant given blood meal. As shown, the plant given bone meal presented the most growth. The plant without fertilizer did grow faster at first, but the growth began to slow. The plant given blood meal showed the least amount of growth. So the discussion, the results concluded that bone meal fertilizer, which was the fertilizer higher in phosphate, prompted faster plant growth. The blood meal, or nitrogen fertilizer, led to the least amount of plant growth. The data did not support the hypothesis because the nitrogen fertilizer prompted the smallest amount of growth in height. Figure 1 shows that the plant without fertilizer is growing faster than the plants with fertilizer, but the growth began to slow. It shows that the bone meal fertilizer resulted in the most amount of plant growth. The limitation of this experiment is that it was only conducted on tomato plants, so the conclusions can only apply to tomato plants. A broader implication is that this experiment can be reconducted with other types of garden plants. More types of fertilizer could also be used to experiment. An error that could have occurred in this experiment is that the bone meal fertilizer also included calcium, while the blood meal did not. I conclude that the bone meal fertilizer, which was higher in phosphorus, is a better option for faster tomato plant growth, as compared to blood meal, which is higher in nitrogen. If someone wants to grow tomato plants faster than just using potting soil, they should use blood meal. This conclusion can only be drawn for tomato plants since it was only conducted on tomato plants. These are my references. Thank you for listening.